single state given, then there is at least 2 hours difference between the East and West.
Yeah, got it.
Mostly because of school drop-off pickup. So it's not too early or too late.
That makes sense. All right, Julie. We are now approaching 3:05. You want to start us off?
Yeah, let's get started. So welcome to our first virtual PCDH19 Town Hall, which is a partnership of UCSF and the PCDH19 Alliance. I wanted to thank the researchers, clinicians, and our pharmaceutical partners who have generously volunteered their time to not only be here today to speak with our families, but who have given generously of their time over the last several years to help the Alliance make progress and serve our families to the best of our abilities. We cannot do what we do and get where we are going without the folks on this panel. I wanted to also give a special thank you to the members of our Scientific Advisory Board, and especially to Dr. Jozef Sullivan for organizing this town hall to ensure that we had a way to engage with our families until we can safely meet again in person. Thanks for being here.
All right, thank you, Julie. So I'm just going to share my screen here. So hello, everyone. My name is Joe Sullivan. I'm a pediatric epileptologist at UCSF, where I direct the Pediatric Epilepsy Center there and have had the privilege of working with Julie for now going on 13 years, I believe. I'm her daughter, Violet's physician, and it really has been a really rewarding partnership to have with her and the Alliance as a testament to everyone who is here today. So not to sort of crack the whip right out of the gates here, but I do, these Zoom meetings can sometimes get a little bit unruly. And so having done a fair amount of these, I just want to go over the sort of format and ground rules, so to speak. We just ask that you please keep yourself on mute. And for this first part of the town hall, keep your video off so that the speakers can all actually be seen more easily on your screen along with their slides. The format is then going to be 5 speakers are going to give us sort of 7-minute, what we call an elevator pitch style presentation. We're not going to go on ad nauseum. We do that when we have an all-day in-person session, which we again hope to have soon. And then after all of the presenters have had an opportunity to give their presentations, we'll have everyone turn their video on so we can try and simulate an in-person meeting as much as we can. But we still would like to ask that you type your questions into the chat. I think that'll allow the things to proceed in a little bit more orderly fashion. And what'll happen is I will read through the questions and unless someone it's clearly for them, I'll try to sort of designate who should take the first stab at the answer. And we know you all are certainly you're here because you want to improve the lives of your children or your loved ones who are affected with PCDH19. And certainly, asking broad questions that may relate to your specific situation is okay, but we want to try to avoid having specific questions being asked about your child because we know that every child is different and we want to just give everyone a chance to be heard. So with that, here's sort of the list of speakers. So I'm going to go first here and just give a very brief overview of PCDH19. Then we'll hear from Dr. Jozef Gécz about genetics overview and update. Then we'll hear from Dr. Paul Thomas about an animal model update. Then Dr. Ann Poduri with her animal model as well as a registry update. We then have Dr. Alex Aimetti from Marinus here, who can give us some of the exciting results from the recently completed PCDH19 trial. And lastly, we have Sharyl Fyffe-Maricich, who is a new board member for us at the PCDH19 Alliance and works at Ultragenyx, and will sort of introduce us to Ultragenyx and sort of their approach to rare diseases. So without further ado, this kind of summarizes as best I could the history of PCDH19 in one slide. So 40 years ago, 50 years ago, am I doing my math right? 50 years ago, I should know that since it's my wife's birth year. A large multi-generational family was reported with a convulsive disorder and mental retardation limited to females. I know we're no longer using that term, but from a historical perspective, that is how this family was initially reported by Juberg and Hellman. Then 26 years passed, and the same family, the gene was actually found to be mapped to the X chromosome in 1997, and that was reported by Steve Ryan. Then fast forward another 11 years, one of our speakers here, the PCDH19 gene was actually discovered by Jozef Gécz in his lab. And shortly thereafter, in the following year, the PCDH19 mutation was found was found actually in an affected male, and then that's also a very interesting story that I don't have time to go into. And together, once the affected male was found, some of the leading hypotheses as to how did this sort of genetic variant or mutation end up causing the actual phenotype or the disease that is epilepsy, but this really allowed this cellular interference hypothesis, which we'll hear a little bit more about today, to come into to final sort of acceptance. Since then, there has been a lot more work that has gone into trying to understand why our patients are having seizures. And there's some studies that have actually been shown to have this sex bias gene expression, again, mostly by Jozef's lab. This has actually led to a few clinical papers that have come out of a few groups that have actually shown that there is reduced steroidogenesis in PCDH19 patients. And then ultimately, just a few months ago, the first clinical trial in PCDH19 epilepsy called the Violet study released their results, and we're going to hear more about them at the end. So, 50 years is a really long time, but I hope my colleagues would agree. We are in a different era now where hopefully, maybe this is the calling, our pace at which we are going to make scientific progress is not going to go in these 20-year gaps like we had in the very beginning. And I know there are a lot of families here that are very well read about the clinical presentation of children with PCDH19, but for those who maybe have just been more recently diagnosed and maybe are a little bit new, I thought I would just summarize what we clinicians call the phenotype, which is really the clinical picture of how these kids present, and therefore when we as clinicians really should suspect this diagnosis. We've come a long way with regard to, I think, making this diagnosis, and a lot of that is due to the availability of genetic testing, where we can now get genetic testing in young children who are presenting with their first onset seizures. And thankfully, even if someone doesn't have the sort of clinical wherewithal to say, oh, I think this is a presentation of PCDH19, they may get a diagnosis by accident, which I think is still a good thing, right? Because then when the treating physician gets that genetic report back, they can hopefully approach their treatment in a more syndrome-specific way and put you in touch with organizations like the Alliance. So this is the largest study reported of PCDH19 patients, so 112 patients, 15 of whom were male. You can see here that the presentation, the age of their first seizure is fairly wide. And when I first started taking care of kids with this, we kind of felt maybe it was 18 months where you had to present. But I think as we have gotten further on, we see that age range can be out to 5 years of age. But the median really is. So the most common time is in that first year of life. The hallmark of PCDH19 epilepsy are these clusters of seizures. And there's some very nicely written papers who talk about seizure clusters in a quote unquote sort of stormy onset. And so that is really when we do suspect this diagnosis. We've also noted that seizures, while they can be of multiple different types, they do tend to be focal, meaning they start in one area of the brain when we capture them on video EEG. They often are triggered by fever, and they often have these affective features. So whether or not they be sort of screaming or sort of a feeling of fear, and that does tend to be, again, not in everyone, and everyone is different, but this does seem to be, a fairly common seizure type in our patients. This is the statistic I think that so many of our families kind of look to and hold on to and wish it was higher and want to know if it's going to affect their child, because there have been observations that about a quarter of young adults or teens will actually have what we call seizure offset. Doesn't mean the seizures completely go away, but meaning they don't tend to be as frequent. And again, you can see the age range here is very, very wide, right? So it can be as young as 11. And I should mention that this was a retrospective study, right? So it's going back and asking patients and families about each individual that was affected with PCDH19. So the range can be very wide, again, with this median around 17 years. And then when we look at those, when this group looked at that subgroup, those were, the majority of those had been seizure-free for 10 years, but actually only 5 of them had actually been off of medications for 5 years. So, we talk a lot about what defines epilepsy remission. Does it really mean off meds? Is it on meds? But hopefully we would all agree that being seizure-free for 10 years is a desirable and clinically meaningful outcome. And then lastly, this group also looked at some of the non-seizure related factors that we know are so important to our patient community, and did note that there was a trend. Again, this is not true for every single patient, but that there was a trend with worsening cognitive and behavioral outcomes if you had earlier seizure onset. So I'm just going to quickly go through some of those, some of the figures from this study. This is basically a figure that shows on the top row, patients who have experienced seizures, cluster seizures, isolated seizures, or status epilepticus at any time. And then the second row here is showing those that actually had those seizure clusters, isolated seizures, or status epilepticus within the last 12 months. And so again, you can see clusters are the hallmark of the presentation of these patients. But then you can see that if you really look at, okay, in the last 12 months, not everyone, it's about half there, right? About half have not had a seizure. And this actually, I think, is a good thing in a way, but it also plagues us as clinicians and families, because they just wonder, okay, I've gone a year, when is the next cluster going to happen? So that uncertainty is really problematic. Here is a figure showing the different non-seizure related issues that affect our patients with PCDH19. And I will say that this is not specific to PCDH19. This is basically a childhood epilepsy issue where we know that seizures are just the tip of the iceberg, and we care very deeply about all of these other, what we say are cognitive and behavioral comorbidities. And so you can see intellectual disability on the left, autistic spectrum disorder. I'm not going to read through them all, but you can see that they do affect anywhere between 30 upwards of 80% of the patients in the study, ranging in severity from mild, moderate to severe. And again, these were assigned this whether or not it was mild, moderate, or severe by the person who was answering the questionnaire. And then lastly, this is a different study that actually looked at another group of patients to try and see how are they responding to our different anti-seizure medications, right? Because in clinical epilepsy, we feel that if we can make a syndrome diagnosis, then maybe we can look through a different lens and maybe figure out what medications are going to be best. I hate to be the pessimist, but really there is no one size fits all approach for these patients. What we can see here is that after 3 months, clobazam, that's the CLB, and bromides, the oldest anti-seizure medication that we have, were the ones that had the most favorable response. And you can see this is 3 months. So this also gets into, how many patients are actually in natural history are just going to have 3 months of seizure freedom without any treatment change. But then if we look at the 12 months, again, we can see that the number of patients that we're looking at here in that sort of dark teal that were seizure free for a year was almost 20% in those that were on bromides, as well as clobazam, and then valproic acid, which is Depakote. So, but hopefully what you can see here is that there are, this dark teal or even the lighter teal where we're getting 100% response, which is seizure free, or 75% reduction in seizures. You can get some of those outcomes with all of these different medications. And then the last thing I'll say about this is the red is the aggravation. I'm not convinced. I'd be curious if Anne agrees or disagrees with me. I'm not convinced that these drugs are necessarily aggravating like they can be in other seizure syndromes. Rather, I feel that this is just showing that they're probably not super effective. And so you're adding in a medication when things are very, very stormy and the storm continues. And so therefore there can be sort of this perception that when I added that medication in, things actually worsened. And I just say that because I don't want to scare folks away. If any of your children are on these drugs and they're doing okay, that's okay. So in summary, I think that the reason why we're all here today is that there still is a huge unmet need for this patient population. There still remains no single go-to treatment approach with regard to maintenance background anti-seizure medicines, or in my opinion, even for rescue medications. In my own patient practice, I have some that respond to X, some that respond to Y, some that respond to Z, and that's not the same across all patients. We know that there's a lot of variability in these non-seizure related comorbidities, such as developmental delay, autism, and psychiatric and mood disorders. The underlying pathophysiology, which we're maybe going to hear a little bit of an update today, is still under investigation. Furthermore, while it's exciting that the first clinical trial in PCDH19 was completed, it was a challenging trial. And I think we'll see a little bit, Alex will show us a little bit that due to the variability in the seizures that just happen in these kids, namely the longer intervals. I just want to sort of give a nod to the frustration of our families that it seems that there's not enough research going on or enough clinical trials, and why is Dravet syndrome and Lennox-Gastaut syndrome getting all the attention? I hear you. I can tell you that as in the pediatric epilepsy community, we really are trying to partner with industry sponsors that may have medications or therapies in development to try and determine whether or not that would be appropriate to study in our patients as well. So with that, I will stop sharing and I will turn it over to Joseph.
Thank you, Joe, and thank you, Julie, and thank you everyone for having me. I have a few slides. Hopefully now you can see them. As Joe mentioned, we were lucky to identify the cause of this disorder back in 2008. So it's 13 years. It's a relatively short time, which have been fantastic to go through, but also frustrating that we haven't really reached all the answers we'd like to have. I'm sitting here in Australia, in South Australia. That's where Paul is as well. And if you hear a little bit of accent in me, that means that I'm originally actually not from Australia. I'm from Slovakia, from Central Europe, but I spent last 27 years here down under. So when we're talking about PCDH19 clustering epilepsy, and you may have noticed that we went through many different iterations of different names for this disorder, but I think we finally settled on this one to be inclusive of the knowledge also we're gaining about what is driving it and who is presenting. So this is still a unique disorder among about 4,500 known genetic disorders and more than 2,000 plus disorders of neurodevelopment. The difference is that here I illustrated it a bit boldly, that you can have either a green central nervous system or purple. And the difference between green and purple is that you have functioning PCDH19 or you don't have functioning PCDH19, and you still can be functioning perfectly fine. In individuals with this disorder, the kind of the brain takes a different pattern, and Paul perhaps will tell you a little bit more about it. And that's what is reflected in that term, cellular interference, cellular mosaicism we refer to. What is really intriguing that, of course, the green brain can be in normal boys, which are squares, and girls, which are circles, or we have these boys who don't have PCDH19, and which we know they are not affected. And we assume, but we haven't come across such an individual, of a girl which would have both chromosome X's with is a variant in this specific gene. And then we know that the girls who have one X chromosome with a variant in this gene and one normal chromosome, or with the wild type, we call it variant, are usually affected. And of course, the boys who acquire error on the X chromosome in some cells after fertilization are also affected. So we were really intrigued by many questions, but I'm really touch on a few. So as Joe alluded to, is this just a seizure disorder? Because that's what's in the name, but we really know it's not. And that's very important for many different reasons of clinical trials, intervention, for us to understand what is driving. It is the same thing which is driving all different outcomes, or it's more complicated. Then we also wanted to know the boys who are affected, so-called mosaic boys, are similarly affected as the girls. So we structured the survey that way. And are these boys who have these mutations so-called transmitting boys? So these are the men or boys who have these variants in every single cell. Are they truly unaffected? And then, of course, we wanted to know, we all have changes in our DNA. So how do we know that this change in PCDH19 is really responsible for the clinical presentations, which is very important for many different reasons, that we really focus on the right disorder with our tools. So this is some time ago, my PhD student, Christy, has done this study when this is a graph where on the left-hand side from 6 o'clock to 12 o'clock, you see the age of onset. So it's before 12 months or after 12 months of the first seizure. And on the right-hand side from 12 to 6, you can see the outcome in terms of the cognitive, intellectual learning outcome. And generally, we found out that if this first seizure happens before year 1, usually the outcome is a lot more severe if the first seizures happen after the first year. So that's perhaps useful, but we still need to figure out why this is the case. But what that also led us to look at on some of these variants, like we have this PN340S, which we see in more than 20 individuals, and this other one, which we see more than 30 different unrelated individuals. And you can see that individuals with one or the other, green or purple, can have seizures very early in their first year or even after the first year, and the outcomes can be very variable. So what this means that even if we have the same damage to this molecule, we really can't predict how early the first seizure happens and how severe outcome we're going to see. This is potentially a problem because genetics is not only about diagnosis, but also about prognosis, and for us knowing what to do and how to do. But this also tells us that even if we have identical twins, monozygotic identical twins, and there are more about 6 pairs like this with PCDH19 variation, one can be perfectly fine and the other one is typically affected with this clustering epilepsy. Now, Joe mentioned this survey, but this is a different view of it. And here we measured, of course, the different other neuropsychiatric, neurocognitive aspects. And here are the scores on the y-axis. But what we see and what this told us was that the mosaic males and the heterozygous females are really similarly affected. So I had the scores in a similar band between 17 and 60, 60 and 70. So they look like they are similarly affected for the purpose of the exercise. There might be nuanced differences, but we need larger cohorts for that. What was also interesting from this, that the transmitting males, so these are the males who have the PCDH19 variant constitutively in every cell, and the heterozygous females, but who are not affected, are truly also similar. And doesn't want to go back. Are also similar, so unaffected. So this is a different way to assess the effect of this variant on different aspects of neurocognitive behaviors. Of course, we looked at a lot of these individual variants, and team member Yuyan looked at more than 322. Here they are piled up on a cartoon of a gene at the beginning of it, and you see there's many of them, and one part of a gene is a lot more abundant than the other part. But in a nutshell, what we managed to do with respect to the population variation, which is not causing any harm, we found a statistical measure or a way how we can tell, or tools, that a particular variant is likely to give a presentation of clustering epilepsy. And I think that's very important for the future, for the proper diagnosis of these individuals, and also for the enrollment in clinical trials, like Alex is going to tell us. So this is a good outcome. This is a study which is currently being reviewed by our colleagues, and hopefully we'll be able to publish it and disseminate it. So it's used more broadly and across the world, which much better and more accurate assessment of these variants when they are found. Because that happened also in PCDH19, that we had some parents who participated into the Alliance because they received a test which suggested that they have PCDH19 clustering epilepsy, but at the end, the variant didn't pass the criteria and was considered to be not relevant to this. So some of the take-home messages, yes, PCDH19 probably is disposable for boys, and likely probably for girls also. That's still a question, because we know that these, at least the boys, don't look any different to other individuals. The mosaic boys, which are the postzygotic, so these are the affected male, are similarly affected to the females. But again, we only have about 17 of these altogether. So maybe small numbers with respect to the hundreds, up to 500 of the girls. We know that this is not just an epilepsy. The seizure is a good predictor of the outcome in neurocognition and behavior. And we have very good tools to assess this variant now, which I think it's a very important step into so-called precision medicine and the clinical trials for this disorder. So, and these are the people who really contributed to this work, and you will hear from Paul in next couple moments. So that's all from me. Thank you very much.
Thank you, Joseph. It's great. Paul, you want to take it from here?
Thank you, Joe, and also Julie and everyone involved with putting this together. I think it's a fantastic event, and certainly something we can do during these more difficult times to get together, and hopefully it's going to be valuable for everyone. I'm sure it will be. So as Joseph mentioned, we've been working together actually since Joseph identified the gene back in 13 or so years ago, and got me involved in the study. So just briefly, I work in this building here. It's called SAMRI in Adelaide. So I'm in the same city as Joseph, and in fact, Joseph's building is just a couple of 100 meters down the road from mine. So we work quite closely together on this project. I'll just get that full screen. There we go. Yeah, so as Joseph's already taken us through, this is an unusual disorder to work with because in these affected individuals, they have 2 X chromosomes. If they're a female, one of them contains the variant, as Joseph put it, or mutation, the change in the DNA, and the other chromosome is normal. And this is a very unusual genetic situation. And essentially what my group has been trying to do is to understand how this unusual genetic configuration, if you will, affects the way that the brain develops during both embryonic development and also after birth. Why is it that we see such variability in the patients that both Joe and Joseph have described? What types of brain cells are affected? So when the brain develops, I don't know why that's flipped forward. I don't know why that's, okay. Sorry, I just have to get out and go back in again. Okay, I can't seem to go backwards, and hopefully it won't flip forward again. But we're also interested in understanding when and what types of cells are affected in the brain, whether it be neural stem cells, so the very early brain cells, or the neurons that eventually form from those stem cells, and whether there's any possibility of reversing the effects that we see, the changes in the brain, whether it's possible to go back, if you like, and change the genes, the genetic configuration, and whether that can have an impact on the way that the brain will function as a result of that. So that's essentially what we're interested in. This slide is just to give you a feel for the approach that that we have been using over the last few years. This is not predicate here in 19 epilepsy, but rather a different syndrome, but it just makes the point that individuals that have genetic changes, we can model those in mice and effectively make the same genetic change in the mouse version of the gene. And this will often give you a very similar so-called phenocopy. In other words, the symptoms that develop in the animal model are very similar to what we see in the affected patients. And this is a useful way of understanding what the underlying cause of the genetic condition is. So we use mice quite a lot for this type of research. Oh dear, it's frozen. Okay, sorry. I haven't had this problem before ever, so we'll just have to.
Maybe it's a Northern Hemisphere, Southern Hemisphere conflict.
Or it's Sunday morning and it's just not waking up for me. Okay, let's try again. Okay. So we know a little bit about what protocoherin does as a protein. So obviously protocoherin-19 is a gene and it makes a protein, and that protein effectively acts like Velcro, if you will. It's if you can imagine a cell as an orange, then the protocoherin-19 protein sits in the skin of the orange and pokes out to the outside. And as shown here on the left-hand side, it helps to stick cells together. So we know it has an important role in helping cells to associate. So one of the earliest things that we did is we looked quite carefully during brain development to see exactly which of the cells express protocoherin-19. And probably not too surprisingly, we find that it is present in many, many cells of the brain, both during brain development, so in the stem cells, in the early neurons, and also in the mature brain itself, and you can see that here. So this is mouse brain sections, but we also know now from human studies that it is present within the developing human brain and mature human brain as well. So the next thing that we did was to try and get a handle on what it is that, what's the effect of having the mutation on one chromosome, X chromosome, and not the other? How does that affect brain development? And the answer to that is shown here, and this harks back to this cellular interference hypothesis that you might be familiar with. Essentially what happens is that the mixture of having two different cells, ones that have functional protocoherin 19 and ones that don't, causes the brain just to develop differently at a very early stage. And that's shown here by these zebrafish-like stripes that you can see in the developing brain. This is what the pattern would be under normal circumstances, and this is what it is in the protocoherin 19 phenocopy in mice. So there's very strong evidence that protocoherin 19 is having an early impact on brain development in the way that the stem cells associate with each other. And then further to that, when we have a look at the brain function in these animals, once they've fully formed and they've become adults, we can see that just in these heterozygous mice, this is the correlate, the genotypic correlate of the affected girls and boys, you can see that the activity is much higher, as you would expect if there's an underlying alteration to the circuitry in the brain that's consistent with the epilepsy that's seen in the affected individuals. Now, one of the things that we've been really interested to look into is why is there such a variation between different individuals? And I think the answer to that really lies in the fact that every brain in a female is unique, because the way those two X chromosomes behave causes them to be unique. And what happens during brain development is that in every cell in the brain, one of those X chromosomes gets shut down. And that's essentially a random process. So if we have a little look along the brain samples here, these are all different brain samples, but they all are have the same genetic makeup, if you like, of the affected individuals. They're heterozygous, and you can see that the pattern of the zebra stripes is different in every one of them. So I think what that's telling us is that the variation that we see amongst the spectrum of patients that have this condition, it's at least partially reflected by the fact that the pattern of X inactivation in the brain is causing different stripes to occur during brain development. And that's something that we're very interested in. Just lastly, I'll show you a couple of sort of recent bits of information that we've been gathering. So far, I've shown you that the protocoherin gene is important during early brain development in the way it affects the neural stem cells and the way they come together. We've recently discovered that protocoherin is actually also present on the mature neurons that are generated in the brain towards the end of brain development. And in fact, we're now starting to look quite carefully at what happens when you just look at the function of protocoherin 19 in neurons themselves. And again, we're seeing some unusual effects when we try and recreate, or when we do recreate the situation that we would see in the brains of affected individuals. So I won't go through too much detail here, except to say that these two are controls where the cells are uniform in their protocoherin 19 status. And here we've recreated that mosaic mixture of positive and negative cells, and we see quite unusual effects that again affect the way that these neurons talk to each other. So to summarize all of that, when is the brain changed in development with a protocoherin 19 heterozygous genotype? Well, we think this is definitely occurring before birth. At least that's what the mouse models are telling us. We're telling us that the way the neural stem cells talk to each other is probably different. And it may also be occurring after birth in the way that the neurons talk to each other in terms of making their connections. Why is it such a variable condition? Well, as I explained, this X chromosome inactivation process is random, so each brain is unique and will therefore have a different configuration of positive and negative cells. How that plays into the exact phenotype of each individual is a very complex question, and one that we do need to look at further. What brain cells are affected? I think basically it spans the spectrum all the way from the earliest stem cells to the neurons themselves, probably not all the neurons, but a decent proportion of them. And lastly, what we're trying to do now is some experiment to see if we can reverse the symptoms. So what I mean by that is what we're trying to do genetically in a study is to create the protocoherin 19 genotype at different stages during the brain development, and also change it such that we get rid of the mosaicism at different stages during development. And that will enable us to, for example, tell us whether it's important that the the neural stem cell stage, is that important for giving rise to the ultimate phenotype in the individuals, or is it more about the neurons and what happens after birth? So they're the kinds of questions we're trying to get to at the moment. And I'll finish up there and just say thanks to the people that really do the work in my lab, particularly Daniel, who is now actually sort of in a nice loop. He's in California doing a postdoc at Stanford. Sandy is great. She's made all the mouse models, and Steph is the current postdoc who's been working. Thanks so much, Joseph. It's been a great collaboration. We continue to work productively together, and particularly thanks to the Alliance for their support for this project. And I'll leave it there. Thanks, Joe.
Thank you, Paul. And I want to encourage attendees you can actually start typing questions into the chat as they come up. We'll get to them after all the presenters have gone, but that way we're not having the onslaught come in. So if a question comes up, it'll also give our speakers some time to maybe think about an answer. So with that, we'll have Dr. Poduri go next. And you want to take the screen?
Okay. Always hard to follow Joe and Paul because of their beautiful data and beautiful descriptions of how we are conceptualizing these disorders. Can you guys see all the little pictures of the people on the side here, or do you just see my slide?
I think it depends on how people do it. So you can, I can see your slide, and then you can drag the gallery to be as big or small as you'd like. So
move Joe's head over to the corner. Okay. So I'm really pleased to be here. Thanks to Julie and Joe for inviting me, and I'm excited to present very briefly some research updates. This is mostly going to be work done by Lacey Smith, who many of you know, who's been involved in our registry. Christelle Ashkar has been a clinical colleague who's been seeing many of our patients in our program with PCDH19 variants. And then Barbara Robbins is a postdoc who's doing essentially all of our zebrafish work now with Tina, who's one of our research assistants. So this is a group effort, and I get to be the messenger and present some of the data. I just wanted, before we get too far, because I know I'll go long, I wanted to make sure to just acknowledge the Alliance for all of your support, both moral and in terms of funding, getting our registry going with UCSF and BCH, which has really been the crux of a lot of the work I'm going to talk about today. So we use an overall framework to talk about many different genetic epilepsies, but I just want to make the point that our focus obviously here tonight is PCDH19, but it's been the focus of our group for reasons I'll get into. And it's really sort of led the way in terms of how we think about integrating our really intense care and passion for the clinical work that we do, which is kind of represented in the inner circle here, diagnosing kids with epilepsy, doing genetic evaluation, and then clinically treating, which is largely empiric, meaning trial and error. Integrating that with this outer circle, our research, whether it's clinical research over on this side or bench research over on this side, hopefully leading eventually to precision therapies for disorders like PCDH19. As we look at the different parts of this puzzle that it really should all be fitting together, I'll mention that it was the work from Australia and the early descriptions of the syndrome, and then Steve Ryan's paper describing the gene, Joseph's work really telling us more about the genetic underpinnings of this disorder and the mouse models that have ensued. That really has, over the years, been driving our push to make a genetic evaluation in the clinic. And so early on, certainly in Boston, not just at my institution, but at others, this gene was on our radar from all of these colleagues telling us at meetings. They look for these girls in particular, now we know also some boys, but who had clustering seizures, who maybe might have been mistaken for Dravet syndrome early on, but have this unique clustering aspect that should make you think about PCDH19. And that's why my group got interested in this long before I had a wet lab. We were seeing patients in our clinical program, and we started testing more often for PCDH19 and got involved with the Alliance a bit to try to understand how can we do better for this unique group of patients who have grown now over the years. That led us to start the registry that I'll tell you a little bit more about, along with Joe and Nilika Singhal at UCSF. And this has really been a tremendous way to really figure out what it is that we want to model in our models. What are the key core features, the really key fundamental aspects that if we're going to make a translational model that we really want to be able to capture and recapitulate. That then led to my choice of PCDH19 as the first gene we started to model in the zebrafish, which is the model that I chose, really based on Scott Baraband's great work looking at Dravet syndrome and how that could be such a lovely way to not only look at neurobiology, but also potentially do high throughput screening. I've presented some of that work in the past. I'll give you an update today. As you may notice, the years have gone by, and it's certainly a much harder and more complicated problem to tackle than perhaps I thought it would be when we started all of this, but it's been very rewarding, in part because these different aspects are synergizing and we can learn from them together. I won't talk about precision treatment, but you're going to hear about clinical trials in a little bit, but I will mention also that we, particularly Lacey Smith, has been involved with ClinGen and efforts to really do expert variant review on a case-by-case basis. And that's going to depend not only on all of the work you're hearing about today, but also on specific variant modeling and looking at each variant individually in the clinical context, but also in the experimental context. So a lot of work going on there and to be done. I'll mention that, as Joe and Joseph and Paul have mentioned, in patients, we think about seizures, but also non-seizure effects, cognitive effects, behavioral dysfunction. And because we know that these are such prominent core features in our patients, we also want to model the seizures, cognitive effects, and behavioral dysfunction in our animal models and look for some of the really core fundamental aspects, hyperexcitability that gives you seizures and maybe gives you some of these other aspects in not only animal models, but potentially cellular models, but also structural defects. And I'll quote a colleague of ours, Amy Brooks-Kale, who says, we use the animal models not because the humans don't tell us what we need to know and what to focus on. The patients do tell us what we need to know and what to focus on, but we can do experiments in animals that we would never do in humans. We really need to have these models in concert with our patients to be able to really understand this disease better, ultimately leading to better treatment for our patients. So in terms of the clinical research we're doing, this is work done largely by Lacey Smith, and I'll just put in my plug for help right away, which is that if you're interested, we could use your help. Our registry that we've done in partnership with Joe and Nilika at UCSF has now enrolled 70 probands and 10 parents. What we have come across is a good phenotypic description, similar to what Christy and Joseph have described with these key core features. We're hoping now to keep going, putting all of these data together to look at severity and genotype-phenotype correlations, inspired in part by the paper, the Poduri et al. Paper that you've heard about from Joseph and Paul. We've been trying to look now more systematically at MRI features in our patients. Most of our patients were presented to us with normal MRIs, but we're really trying to look at now deeply and take a relook at the actual raw data to see if that's really the case. The other thing that I'll mention is through our registry and collaborations with Nilika. Nilika's been really looking at whether patients with PCDH19 variants should be considered for epilepsy surgery, particularly if they have focal epilepsy. So that's something we're happy to be able to collaborate with Nilika and Joe on. The last part where we really need help is with some deep phenotyping. We have the ability to work with a neurobehavioral core to look at patients and their parents, including variant positive and variant negative parents. And the hope there is to try to look more deeply to see if there's specific types of speech disorders, specific types of autism spectrum disorders that might be uniquely present in our patients with PCDH19 variants. We also want to look at the parents for a couple of reasons. We only have 4 males in our cohort, so we don't have a lot of boys, but we do have some variant positive fathers and variant positive mothers. And we thought it would be important to look at those individuals to see if there might be some maybe subtle features that haven't yet been characterized. The reason for that, I'll get to in a minute, is just really particularly looking at the dads is, are there some specific and very small in the grand scheme of things phenotypes that we've been missing because we've been looking for clustering seizures primarily? And are there some perhaps non-mosaicism dependent features that we might see in either patients or parents that we might be able to then model in our animals? So in terms of the translational research, this is all work now done by Barbara Robbins. We've been doing a couple of things. We've been trying to create something in the zebrafish, which we've chosen again because it's a nice high throughput model. When we get to the point of drug testing, we're not there yet, but we've chosen the zebrafish for that reason. But we acknowledge that we can look at mosaics. We can actually create mosaics in a couple of different ways. But it turns out that zebrafish don't have X chromosomes. They have this Z chromosome, which gives you male versus female. But at the age when we can study seizures and behavioral disorders that are really very, very tractable to do in 96 fish at a time, the fish don't actually display any dimorphism, meaning we can't tell if they're male or female. And in fact, the PCDH19 gene is not located on an X chromosome like chromosome. It's it's autosomal. It's like any other non-sex chromosome. And so in that way, what we can do is create several different experimental setups. We can create mosaics by injecting at a 2-cell stage, or there's all sorts of different molecular ways we can make mosaics. But we can also look at heterozygous and homozygous fish that basically have their own value, not recapitulating the human genetics, but looking at what happens if half of the amount of PCDH19 is present but abnormal in all the cells. Or what if you just have no functioning PCDH19 at all? If you recall, Joseph mentioned we don't have any known female patients with both X chromosomes affected, with both PCDH19 affected, but in an animal model like this fish, we can actually do that quite readily. So that's been something we've been wanting to look at, in part to see, do you really need it at all? If you can have no functioning copy in a male, one might argue from first principles, well, maybe it's not even necessary. We know it is necessary. We know it's an important gene. And we've used the zebrafish to try to isolate these different conditions. So what have we seen? I'll go through this quickly because I know we only have a few minutes, but we're hoping to put some of these data together for a paper relatively soon. When we look at zebrafish electrophysiology, and this is putting an electrode, it's not an EEG, although we can do that as well, but it's putting an electrode into the head and into the brain region of the zebrafish. If you look at the wild type, this is just a normal fish. There's a certain amount of activity, and I know in years past we discussed with some of the other researchers, why is there this much activity, and they wondered if there shouldn't be a flat line rather than some activity. And what we discussed then and will maintain now, and has been borne out by Scott Baraban's data and others, is of course there's some activity. They're 5-day-old larval zebrafish. They do have brain activity, but what happens when we remove one copy of the PCDH19 gene, so every cell has one missing copy, is we see all these bursts, boom, 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 all these bursts over and over again. This is a 10-minute scale, so it's a few bursts per minute. We see this in several fish. And if we remove both copies, in fact, we see similarly more bursts, and certainly a lot more than we would see in a normal fish. We've done this in a couple of different setups with exon 1 being removed, and actually we're introducing what's called a frame shift and removing the whole gene after that. We can do that a little bit further along the gene and have the same thing follow through. And likewise, we see that in a mosaic model that we've created. So this is sort of curious. It's not necessarily what we expected. We thought we'd see something in the mosaic and not in any of the others if this was going to exactly copy what we see in the human female situation. But this is intriguing. It makes more models for us to look at, but in some ways it raises more questions than answers. It seems like there may be some, at least in the fish, non-mosaicism dependent features of this gene. So that's food for thought there. We don't see a ton of seizures, but we do see some. We see some when we introduce a chemical called PTZ, which is a proconvulsant. Our normal MO is to sort of try to suppress seizures, right? We use all sorts of the meds that Joe mentioned, but in animal models, we try to give them a little bit of something that might cause seizures. And we see a little bit, but not a lot, 6% of this one particular line and not a lot in the other lines. So we're not really finding the seizures. And so that suggests that maybe this particular animal model is not as prone to seizures as maybe others, but also that there are other features we need to look at. And we've looked now as a correlate to this. I don't have data. I couldn't get the video to just download from one device to another, but we are seeing that there are other ways to look at hyperexcitability that aren't just related to seizures, it's discrete events. And one of those is calcium imaging that's been used in a lot of different systems. And we are seeing a remarkable increase in calcium signaling in some of our animal models. But again, not just in the mosaic animals, in the non-mosaic animals as well. In fact, our strongest phenotypes are coming from our heterozygous animals, the ones where one copy is missing, but in every single cell. So that's quite curious. It doesn't contradict in any way the nice mouse work that you've seen from Joseph and Paul, but it is raising some questions that we haven't yet found the answers to. I'm curious to see if anyone has any theories. At some stage, because we were getting all of these data that were very, very interesting, but not completely in sync with the human mouse data, we said, well, let's just take a blind look and see, if we look at our knockouts and we look at the wild type, the normal ones, what genes are regulated up and what genes might be regulated down in the fish. It's a technique called RNA-seq where we look at the expression. And we did find some interesting genes that I just want to mention. This is very, very preliminary, just a few animals, but we found some GABA-related genes, GABA-RB2 and GABA-RD, which are related to, this one related to GEFS+, this one related to its own epileptic encephalopathy. And we're just beginning to do some follow-up work to see if this might be related to presence or absence of interneurons in these knockout fish, and so on. So some tantalizing leads here from the RNA-seq. So preliminary conclusions, but really some questions, is that the zebrafish models do recapitulate some of the features of PCDH19 clustering epilepsies, again, in a non-sex dependent way. It's an opportunity to study hyperexcitability. We hope then to be able to study ways to modulate and moderate that hyperexcitability. And it's an opportunity to study complete loss of PCDH19 in a way we can't in human. And from the combination of our human and fish work, we do see some features, overt epilepsy, for example, that seems to be mosaicism dependent in humans. Possibly we thought in fish, but actually we've got some contradictory evidence. Our mosaic fish just aren't seizing. So humans, yes, fish, no. We've yet to be able to explain that. But other features, the hyperexcitability overall, behavioral issues that I didn't get into for the fish, we don't necessarily think are mosaicism dependent, at least looking in these models. And then we have just one father who's enrolled who has a variant, the same variant as his affected daughter, who does have some very specific but notably abnormal features, not seizures and not cognitive, but some restricted behavioral aspects that are quite intriguing. And what we'd like to do now is to be able to look at more. One person is not enough to make a conclusion around, but it is beginning to tell us that maybe there is some value in studying the males who have a heterozygous variant, particularly the dads of our female patients, because there may be some very specific findings that if you don't go looking and digging and have the neuropsychologists do very, very specific assays, we may not be identifying them, but there may yet be some non-mosaicism dependent features, which would explain in part why this gene is truly important, why you're not completely off the hook if you're a carrier male. So more to come, hopefully at the next conference. So with that, I'll thank you. I just want to note that we are now doing this with federal funding, which is amazing, but we got our start with Charles Hood Foundation, the acute syndrome, with some early fish funding, and the PCDH19 Alliance team, not only funding our registry, but actually encouraging enrollment in the registry. So I'm happy to be contacted either through our epilepsy genetics at children's.harvard.edu email, or Lacey's answering the PCDH19 registry email. And both Joe, Nilika Single, Lacey, and I are all really happy to hear from you to see if there's a way to participate. I mentioned on the slide and didn't mention, because of COVID, we've been stuck about doing our ability to do in-person evaluations, which we'd hope to do. Julie, we'd hope to fly you guys out for an in-person evaluation, but we've completely switched gears now and we're doing everything online with online assessments now for the developmental assessment. So I think that will continue in the post-COVID era. I encourage all of you to be in touch so we can beta test that on you if you're so inclined. Thanks very much.
Thank you, Anne. That was great. All right, next we are going to have Alex Aimetti from Marinus, who will give us a summary of the findings from the Violet study. Alex.
Great. Thank you, Joe. Can everyone see my screen? I just want to confirm everything's good. Good. Great. All right. Well, thank you. First, I just want to thank the organizers, Julie, Joe, and the entire Alliance for the opportunity to speak to you all today. I'm going to transition a little bit now from all the great scientific research that we just heard about and talk a little bit about some of the clinical translational research that we've conducted as of late. So what I really also wanted to accomplish was to provide a brief background on who we are at Marinus, our work with ganaxolone in PCDH19. I'm sure many of you are aware of this work, but some of you may not be as well. So I just wanted to make sure that we're all working off the same page. So ganaxolone is an investigational drug, and it's a synthetic analog of a natural neurosteroid called allopregnanolone. And it was some of the seminal work that Joe Sullivan talked about earlier regarding some of the reduced steroidogenesis that Jozef Gécz and colleagues showed back in 2015 that really provided the scientific rationale to supplement with an allopregnanolone-like compound such as ganaxolone. So we've actually conducted 2 clinical trials in PCDH19 to date, the first of which was an open label clinical study of 11 patients, and this was conducted a few years back. And what data from that study showed was that potentially certain patients with PCDH19 may benefit from ganaxolone, and it really established a hypothesis that we thought was worth further testing. And so I just want to show you this graph here. And what I'm showing you on this graph is looking at some of these endogenous or natural levels of the neurosteroid allopregnanolone and the patient's response to ganaxolone. So patients that have a seizure frequency reduction shown here as a negative value, so moving from here to the left, are improvements in seizure frequency. And what we saw was that a larger proportion of patients that had lower levels tended to respond more favorably to ganaxolone. Again, it wasn't 11 patients open label study, so we thought these data were enough to study it in a further larger clinical trial. And it was this result that really motivated us to initiate a biomarker stratified placebo-controlled trial of ganaxolone in PCDH19 clustering epilepsy. And this was the VIOLET study. So just a little bit of details on what the VIOLET study was. This was a study that randomized patients aged 1 to 17 to receive either ganaxolone or placebo on top of existing anti-seizure medications. The primary efficacy endpoint was the percent reduction or percent change in seizure frequency during the treatment portion relative to the baseline period. The study was originally designed to enroll up to 70 patients, but we did back in last year, we had to truncate the study due to some challenges that I'll talk in a little bit more detail about, but we were able to enroll and randomize 21 patients into the study. Some of those challenges that we did run into, some of them were enrollment challenges, some related to COVID, right when we were trying to initiate some of our ex-US sites. That was right around March or April of last year. And then also around trying to, again, when conducting an epilepsy clinical trial and looking at the change in seizure frequency, you have to establish a baseline seizure frequency. And so we were really, again, talking about the clustering phenotype that others have mentioned prior on this call. It's very difficult to establish a baseline seizure frequency due to the waxing and waning of seizures in this patient population. So we were looking for potentially more severe epilepsy patients that had a little bit more of a regular seizure frequency pattern, which is also why it consisted of a 12-week baseline, which is challenging for an epilepsy trial design. It's a long time to be in a study. It's a long time to not be able to change background medications. But following the 12-week baseline, patients would then be randomized again to receive ganaxolone or placebo. And then we're in a 17-week treatment period. And at the end of that 17-week treatment period, we would assess the primary endpoint. So these were data that we released and communicated last month. And we are very anxious to publish these data in a medical journal in the coming months. But what I'm showing you here in this graph is that patients on ganaxolone, 10 patients, experienced a median 61.5% reduction in seizure frequency compared to patients on placebo, 11 patients, which experienced a median 24% reduction in seizure frequency. This result did not achieve statistical significance, and a lot of that is certainly due to the variability and the small sample size. So although we were encouraged by these data, again, we're seeing the directional improvement of ganaxolone relative to placebo. I do want to caution the interpretation due to the low sample size. And just to expand upon that a little bit more, what I'm showing you here is a waterfall plot where we're looking at each of the 21 patients lined up with the largest seizure frequency reduction to the left, all the way to the least seizure reduction, or in this case, sometimes seizure worsening. And what I'm showing you here is where the arrows are pointing down is where those median values are occurring. So again, the 61.5 is showing up right here, and the 24% in the placebo arm. And what I wanted to highlight in this graph, though, is that, again, looking at the difference on the graph to the left, 61.5 versus 24 looks large. There were a lot of placebo patients that had a meaningful reduction in seizure frequency, talking a lot about the variability in the patient population. And again, some of this could be due to the natural history of those patients. So it really highlights some of the challenges in a classic epilepsy clinical trial design. And again, I think, moving forward in clinical development, I think that this certainly could be opportunities for new innovative trial designs in this patient population. Now, I talked earlier about these natural neurosteroid level potentially predicting response, and I wanted to comment on that. I think what our study did was it was the correct experiment to test if this was true or not true. And what I'm showing you here in this graph is that based on the hypothesis we saw, one would expect those with lower levels to have a better response, a higher reduction in seizure frequency compared to those that had high levels. That's not what we saw in this study. Now, do I think that the phenomenon is reversed? No, I think the sample size is too low to make that extrapolation. But what I do think, though, is that the response that we saw in these patients was independent of this biomarker allopregnanolone levels, and potentially more of a response of ganaxolone more broadly. So I just wanted to highlight that because I know it's something that the community may have been aware of in the past. In regard to safety, ganaxolone, although not approved for any indications, has been tested in more than 1,600 patients and individuals over the past decades of clinical development. And so it does have a well-established safety profile, and the result of the Violet study did not show up any new safety signals, in which ganaxolone was generally well tolerated. The most commonly reported adverse event was somnolence, due likely to its GABAergic mechanism. So I just wanted to highlight a few key takeaways from the study because I think it's really important to discuss. The first one was that we did execute the first double-blind placebo-controlled trial in PCDH19 clustering epilepsy. We've already mentioned some of the challenges that we did encounter related to enrollment and then the seizure frequency variability, which potentially led to increased placebo response. With that said, though, we do believe that we have a rich, high-quality data set that we are aiming and looking to collaborate with researchers to publish in various medical journals. And then also, I talked a lot about the seizure frequency, and I do want to say that we did hear the community loud and clear, and we did implement a lot of non-seizure related outcome measures in the clinical study. At this time, we haven't really gone through those data in a lot of detail and haven't reported on those data, but I can commit to that we will communicate those data on a lot of the behavioral scales, sleep scales, and otherwise when appropriate. And although at this time we don't have any future clinical trials for regulatory approval plan, we at Marinus are very open to supporting externally led clinical research in PCDH19 epilepsy and continuing to support the community. So with that, we really want to thank all the patients and families who did participate in the study, and then more broadly, the entire community for the support all along the way. And I have included my email address. If there are any questions, happy to try to address those. Thank you.
Thank you, Alex. That's a great summary of the data, and I'm sure I too am looking forward to some of the non-seizure data that we had all the families keep track of. So last but not least, we're going to have Sharyl introduce herself and say a few words. Do not have slides, so you can relax and take it away, Sharyl.
Great, thank you so much. It's so great to be here, and thank you so much for having me today. Yeah, I don't have any slides. I'm sort of glad because it would be very difficult to follow everybody who's gone today. This has been some really great information and great for me as a new person in the world of PCDH19. So I wanted to take a few minutes to tell you a little bit about myself and about Ultragenyx, the company that I work for. So my role at Ultragenyx is the executive director of molecular and cellular biology. So I work with our research team. I have a small lab, and we basically work to try to understand the biology and do all the preclinical development for our neuro programs. So I can tell you that I've been working on translational research for the last, gosh, almost 20 years now. Doesn't seem like it could possibly be that long. And I've always been focused on neurodevelopmental disorders. I had a brief period of time in my career where I was also focused on neurodegenerative disorders. So I worked on multiple sclerosis for a number of years. But I started off doing my PhD in molecular and human genetics with, of course, a focus on neuroscience. And I did that at Baylor College of Medicine in Houston, Texas. And the focus of my thesis work was a disease called Rett syndrome, which I know most of you might be aware of. It's another X-linked neurodevelopmental disorder. We see it primarily in girls, and it does have an epilepsy component. So some similarities to PCDH19. And for my thesis work, I was really focused on understanding the different subsets of nerve cells and how dysfunction in those subsets of neurons contributes to the symptoms of Rett syndrome. And After that, I went off and decided to do a postdoc and look more at glial cells, which are the other main subtype of cells that we see in the nervous system. And I was fascinated by those because the glial cells actually have an incredible regenerative capacity. We don't see that with neurons. So once a neuron dies, we can't get it back. And you're born essentially with all the neurons that you'll have for your life. So I was fascinated by these glial cells because of their regenerative capacity, and focusing specifically on oligodendrocytes, a subtype of glia that are important for multiple sclerosis as they degenerate. So in 2013, I started my own lab at the University of Pittsburgh. So I joined the faculty there. I was in the Department of Pediatric Neurology, and I had an NIH funded lab for about 4 years. And it was a great experience, a great place to be. I really enjoyed doing that kind of research, but I felt a little bit stuck. And this was certainly a personal experience for me, in that I really wanted to develop medicines for patients with genetic diseases. And I felt like I was doing all this great biology in my lab, and I was writing grants and I was writing papers, but I couldn't figure out how to get to that next step, how to really make a medicine that would treat a person. And for me, that's what I wanted to do. And certainly there are lots of academics who are very successful at doing that. But for me, I just felt very stuck. And I was really fortunate to have an opportunity to join Ultragenyx. And really, my focus now is on bringing therapies to patients, and specifically patients with rare or ultra rare genetic diseases. And it's been an interesting transition. I've been with Ultragenyx about 5.5, not quite 5 years, sorry, just about 5 years now. And I've learned a lot, and it's been different. But I enjoy it every day. I enjoy the interaction that I have with patients, with families, and really just with this very strong mission every day to do everything we do in the lab is ultimately to get that medicine to be able to go into a person and to help them. So it's been really rewarding. What else can I tell you about Ultragenyx? I think that one of the most fascinating things about Ultragenyx is that we take a bit of a different approach. So we focus more on the disease first, and really tackling monogenic diseases. So diseases where there is mutation in a single gene, and we understand what it is, we know exactly what's broken, and it makes it a little bit easier to try and fix it. And that was an approach that resonated really well with me, especially coming from having a background in genetics. And then we have an incredible toolkit. So we're not sort of a company where we just have, we only do gene therapy, or we only use a certain type of small molecules. We basically can do any of it. So we think about the disease first, and we really try to understand that biology. And then we go to our toolkit and we say, how would we best treat this disease? And I think that's been one of the most fascinating things for me today, is learning more and more about PCDH19, is how can I help? How can I help, and how can I use what I know about all the different tools that we have to treat this disease? I can also tell you that we have a preclinical program, which is an AAV gene therapy approach for a disease called CDKL5 deficiency disorder. So you may have heard about that one as well. Another X-linked neurodevelopmental disorder with a huge epilepsy component. And I'm actually the leader of that program. So I started the program with Ultragenyx and have been leading that for the last 2.5 years. And we are still in preclinical development, but we've got some exciting data, and it's been a great learning experience about how to really take an early scientific idea, all the way through, and what does it take? What do you need to do to really develop something so that it's ready to go into patients? And I'm happy to answer more questions about that program if you guys have questions in the next couple minutes. So I'll stop there.
Great.
And
Yes.
thank you very much.
No, thank you. And as I mentioned, Sharyl's on our scientific advisory board and reviews our grants. And so it's always nice. We always, want to advance the science, but as a clinician and as families on this conference, we know that we want to have science that is potentially translatable. So it's nice to have Sharyl's expertise on our board. So thank you. So we have lots of great questions coming in. If for anybody who wants to turn on their camera, we now welcome you to do so we can connect names and faces, and you don't have to. But I will start with some of the questions that came in from the beginning. And the first, I think, couple are for you, Joseph, in your slide about the term PCDH19 is disposable for boys, maybe for girls. Can you elaborate on that? And then maybe you could take the second question too about how does PCDH19 randomly start in a family?
Thank you, Joe, and thank you to Joy Martins and Meredith who asked these questions. I guess I didn't want to say that it's totally disposable, and my colleagues Ingrid Scheffer alluded to it. Maybe these men or boys who lose PCDH19 may have some subtle behavioral issues, but for the purpose of the exercise, they do not have PCDH19 clustering epilepsy, as far as we know. But so that was the statement. Now, why I said that the girls, we don't know if a girl or a woman completely loses PCDH19, what the outcome is going to be, because there are a lot of groups that hasn't been discussed here, but there are strategies to, theoretically, one is to repair the problematic PCDH19 or even shut down the correctly working one, because if there is a precedent that an individual with without PCDH19, which are these boys, can do well, certainly much better than the affected girls, that shutting and completely destroying the PCDH19 could lead to a good outcome as well. At least that's the thinking behind. And that's why I alluded to the fact that maybe girls without PCDH19 are going to be similarly unaffected by the clustering epilepsy as the boys without PCDH19, but that given that they are not the mosaics. There are some questions about this, especially with the recent mouse models, suggesting that maybe the boys and girls may react differently to the loss or complete loss of PCDH19. So if that explains it. Now, PCDH19, how it starts in a family, there are basically two ways. It doesn't start in a family, it just appears in a family because it can travel through individuals who are not necessarily affected. And we know that because the penetrance or the chance that the person is going to be affected is probably about 80 to 90% as far as we know currently. So how it starts, that maybe it was just not noticed in a family and it appears. But it can also start, and these are the boys, for example, the mosaic boys, when soon after fertilization there is error in copying one X chromosome from one cell to another cell, and that error happens to happen in PCDH19, and from that stage onwards, all those cells would have their error and create that mosaicism, which nicely Paul and Anne described as the driver probably of the seizure activity of the brain. So if this answers the question, so it can be in a family, but it pops up, or it genuinely happens as a new error. That's why we see these families coming again and again. So technically, we never probably going to be able to totally wipe out, no matter what we're going to do, PCDH19 errors. We just can diagnose them early and see how we can help to correct.
Great, thank you, Joseph. Seizures in PCDH19. Can you hear me?
Yeah. What was your? You were a little bit sort of lost when you asked the question. Can you repeat it, Joe?
It's switched over. So Anne, do you want to maybe take a stab at that one, or I can take a stab? Can pregnancy take away seizures in PCDH19?
I think the answer to any question about pregnancy is always yes when it comes to epilepsy. The seizures can get worse, seizures can get better. They can stay the same, but I think the reason it's particularly of interest to discuss in this forum is that we know, from Jozef's work and others, that there are effects of neurosteroids in females. And so I don't know, but perhaps Jozef does in terms of the effects on allopregnanolone and from pregnancy.
No, this is a very good question. I don't recall seeing any information or digging into specifically whether during pregnancy we see or would expect protective effect. No, I'm sorry, good question. We need to look for answers.
Yeah.
We will.
It's a great question. I mean, I will say, I think because we have that concern, particularly in some of the young girls and women we have who we expect might actually go on to have their own kids, I think it's an opportunity, because we have this group and we have people like you all asking these questions, that we should look, we should make sure that before childbearing happens, we get some baseline levels of allopregnanolone, but also lots of things. And then at such time, if they're willing to get in touch with us when they are pregnant or about to be, or planning families, so that it would be an opportunity to try to answer that question. Right now we're sort of speculating.
And so I apologize for the video. I don't know, Dawn, if you can work some magic behind the curtain to see if we can allow cameras to be turned on without crashing everything. Sometimes that's the risk, but while we're working on that, so one question came in from Meredith. Have you ever seen mild cases of PCDH19 occurring after 5 years old? I can maybe take the first stab at that, and I'd like to hear maybe Anne's comments as well. I think that with all genetic epilepsies, the more we start testing, the more the so-called phenotype gets broader, right? And so in the paper that I summarize, the 112 patients, I think that as we start to test more and more children with gene panels, it's likely we are going to stumble upon some that are relative outliers from what we would call that classical core phenotype. So it is very possible that there are other patients that may not fit the clustering and may not fit that what we classically see, that may have a PCDH19 mutation, and I think that story is going to be evolving over time. So I personally have not cared for any PCDH19 patient who had onset after age 2. Yeah, Joseph, go ahead.
Yeah, just very quickly, I looked up the answers. I didn't know the numbers, but in that survey, we had 3 individuals. I think it was 115 months, 60 months, and 60 months. The 115 ones was mosaic male who had a history of admission to intensive care unit, but generally the outcome was not bad. The first seizure started at 115, and there were 2 at least who started seizing at 16 months. That's 5 years or around that time, with mild outcome. One with only single admission to ICU, and the other one without any such consequences. But generally, there are very few. The seizure can start up to 7 years. Of the 8 years of age, I think we captured 1 or 2 individuals from larger families, what we had. But generally, the outcomes are not very severe, but it can be severe.
Yeah, I agree. In our paper from a couple of years ago that Lacey had put together, we had 1 child who was 3 at onset and 1 who was 4. They both had missense variants, but otherwise everybody, like Joe, like your experience, everybody was under 2.
Yeah, okay. And then maybe just while we're talking about the clinical, in any other registries, any deaths or comments about mortality? The question was, has there been just studies about overall mortality in the disorder?
I am not aware of that. It'd be interesting to ask Nicola Specchio if he's come across any numbers. I mean, I think what I am struck by, given the numbers that Jozef and Christy had and the numbers that we have, is that thankfully we're not seeing mortality to the same extent that we do in Dravet syndrome, where it's 20% by age 20, and in earlier onset syndromes like migrating epilepsy. It's almost a third of the kids by age 3. And I think I'm quite thankful for that. I quite the opposite. We actually we knew about PCDH19 from all the studies you've heard about. We kind of re-stumbled upon PCDH19 as a field through the Epi4 case study, where almost 4,000 individuals were enrolled and sequenced. And these were mother, child, father, child, or 2 siblings enrolled together. And these almost 2,000 pairs, when we looked to see, what genes are accounting for their epilepsies, and these were often milder individuals, PCDH19 was right up there. PCDH19 and DEPDC5 were the 2 genes that were at the top of that list of what's seen commonly in these otherwise relatively well individuals with epilepsy. At least one of the 2, if it was a parent-child, one of the 2 had to be pretty well. So we don't know for sure, but it certainly doesn't seem to be as bad as some of the other syndromes, thankfully.
Yeah, I would agree. And then, I don't know, Julie, this is always a difficult topic to talk about, but just in members that have joined the Alliance and the Facebook page, have we heard of any unfortunate deaths?
We've 3 that I know of, and one that's in the group that's public. So we have 500 people in the group. That probably represents 250 children.
Okay. Whoops, you're cutting out a little bit, but it's okay. So just a couple of questions about vaccines that I can maybe take. One is, can PCDH19 be triggered by vaccines? So certainly, just for clarity, the genetic mutation cannot be triggered, right? The mutation is there. Certainly, we know that this is a fever-sensitive epilepsy that can present in the throes of fever, and we know fever is a common sort of side effect of a vaccine. Almost tells that your immune response is actually doing something. So it can trigger the first seizures, and in fact, the other study that I mentioned about the mosaic male and how that PCDH19 gene was discovered there was basically, it was being looked at in a group of patients that looked like they had Dravet syndrome, which we know is very vaccine sensitive. And turns out that many of them had PCDH19. So, and then as a follow-up question to that, in terms of vaccine schedule, how do we schedule vaccines for our infants to best avoid triggering a cluster? I'd be curious if Anne has a strategy. There is, again, no one-size-fits-all strategy here for my patients that have shown that they are vaccine sensitive. I will pre-medicate them for 48 hours or 24 to 48 hours with a benzodiazepine, usually clobazam, which is Onfi. So they start the clobazam a couple times a day, the 24 hours prior, and then they continue it for 24 or 48 hours after, just so that there's that little extra protection, acknowledging that it's not 100% to be preventative. I don't know if you have any strategies, Anne.
No, I agree. I mean, I think, certainly the fevers are worrisome because they can trigger the seizures, but we need the vaccines to happen. And what I tell my patients is, that obviously viral illnesses can happen, infectious diseases can happen, as we all know, certainly now with COVID. And I know there's a question about COVID and PCDH19 that popped up. I have had one patient develop COVID from an environmental exposure, and actually shockingly did not have worsening of seizures. But I think, there are preventable things and non-preventable things and knowable things and non-knowable things. And at least with the vaccines, we know what we're doing. We know we're inducing a situation where there's an immune response, and there may well could be, there could be a fever, there could be a reaction that includes seizures. So what we can do is try to pre-medicate and medicate around that risk. And for some kids, that might mean giving them Ativan, giving them something that might be a little sedating for a while, but it's a knowable exposure as opposed to the viruses that we're all exposed to that you don't necessarily have a heads up about. So I think that's how I sort of manage it in my group.
And since you brought up COVID, that was another question as to whether or not PCDH19 children are more susceptible to be getting infected with COVID. And if so, or if they do get infection, are they showing worsening neurologic problems? I think thankfully in the broader pediatric epilepsy community, we've learned a lot in this last year of this pandemic, and thankfully it seems that our our genetic and treatment-resistant epilepsy patients do not seem more vulnerable either to getting infected with COVID, and if they do, they don't seem to spiral kind of like they would with, say, influenza and leading to high-grade fevers. Now, certainly, if someone's symptomatic and has fevers, it could cause them, but it's not something where it appears they'd be more vulnerable. And we've been sharing that with some emails with our colleagues in Italy, and certainly when there have been these sort of surges in pediatric hospitalizations. I'm not sure if you had anything else to add to that, Anne.
I concur.
Yeah, so this is a question maybe for anyone to take a stab at in terms of seizure types. Is there, if you have more than one child with PCDH19, so maybe this will be Paul and Joseph and Anne. If you have more than one child and both children present differently, is there a rhyme or reason or certain seizure types that may or may not become normal into adulthood? And then specifically, there's a question about absent seizures likely to outgrow it as to other seizure types. So maybe I'll just quickly address the absent seizure. I think absent seizures in its purest form are actually rare in this patient population. So that's one thing. But I don't know, just genetic heterogeneity in siblings. Does someone want to take a stab at that?
I think, Joe, the fact that we also see monozygotic, that is genetically identical twins with exactly the same PCDH19 variation, which was passed on, for example, from the father to both girls. And we see that they are discordant and they have their own seizure history and cognitive and neuropsychiatric kind of evolution, suggests that each case is kind of statistically independent, which would Paul's mouse studies would suggest. So I would say while there might be some effect of a similar genetic background, the driver behind it is the mosaicism or maybe something else. And that's going to be really individually specific, and even on exactly the same genetic background. That means also in the family of two sisters, which are not identical twins, but they are just sisters, you would expect, or you may expect, very different kind of history or natural history of this disorder.
I mean, I'd add that we see it in other genetic epilepsies as well, so SCN1A and so on. So it's not unique to PCDH19, but what is unique to PCDH19 is because it's in girls, there may be different numbers of cells that are affected. So you might expect a difference in severity, and which areas of the brain happen to be more affected are going to potentially influence what type of seizure. So it may actually be specific to PCDH19 that there's more variability, but I haven't seen enough sibling pairs personally to know what the range of the variability is, but I think it doesn't surprise me at all given what we know.
I would only just make a quick comment to follow up, and that's that we don't really have a good connection yet and a good understanding of how that variability in each brain that occurs due to that random X activation, how that causes the seizure, what's the change in the brain and in what regions to result in particular seizure types. And we understand, I think, the variability, but we don't understand the direct connection between what's happening here and what seizure type. So I think we have plenty of challenges to overcome in the sort of basic biology side, and we'll try and bring those to translate to get a better understanding of what drugs might be best for different situations. The other thing is that this is an unusual condition to study because each brain is unique. So we need to look at many, many samples to try and get a feel for what's happening. And that is an unusual aspect of protocoderone 19.
Great. Maybe switching gears a little bit, kind of related, is more of a genetic counseling and recurrence risk. So there's two questions. Are there any interventions you can do if you have a baby girl with a 50% chance of PCDH19? So I would assume there,
if they're saying that this always makes my head hurt when I have to think about pedigrees.
I think, Joe, in my experience, we have not seen a case diagnosed with PCDH19 before the seizure onset, and then there is a question whether something going on with the development prior to the seizure onset, and there is only anecdotal evidence that maybe there is some delay prior to first seizure onset. We only recently been contacted by one family where, because of family history, identified PCDH19 variant in an individual very early before the seizure onset. So this is the only one case I know of. And of course, this family was asking exactly the same questions. And the doctor, what we can do, what's going to happen. So in a way, we don't want just to diagnose, we want to have a prognostic markers into the future. And we do a little bit of work. So we have some biomarkers, perhaps epigenetic and gene expression markers, which perhaps might be able to tell us if somebody is diagnosed at the age of 1 month, soon after birth, as part of routine testing, or family, perhaps occurrence of this disorder, that we hope we might be able to predict whether they're going to have severe or not so severe outcome. Now, the question is whether we can do something about it currently, where we don't know whether just stopping or preventing the fever happening, or some other stress or infection, but with or without fever, or just the fever would delay the series, the first cluster or not. Well, nobody knows that, or at least I don't know that that could be prevented, or even if you prevent it, that you prevent the associated kind of changes in development and seizures afterwards.
Mm-hmm. And then there's a similar, and I'm trying to group themes here. So our 8-month-old was just diagnosed. We have a 2.5-year-old girl. How likely is it that she would also have this disease? It's a very interesting question. I mean, I guess I would say that the first step would be to see if dad is a unaffected carrier male, right? And also to test mom and see if perhaps she is a asymptomatic female. And if neither of them are carriers, then the likelihood that the older sibling would be affected would be very low. But now with the availability of genetic testing, you know, I wouldn't be wrong to also test her. This has changed so much now that we don't have to justify getting genetic testing. But I don't know, Anne, if that family was to come to your clinic, how would you advise them?
So I think there's a couple of things. One is, the possibility that one of the parents is unaffected is quite likely with PCDH19, unlike some other disorders. So absolutely, I would start there. The other possibility is that you test blood, and mom, you test blood, and dad, you don't find a PCDH19 variant, and you think everything, it must be just have happened newly in the one child who's affected. But we know now that testing the blood isn't everything, right? So a parent can be mosaic, not in the way that we've been talking about mosaicism with females being mosaic in expression, but that maybe the blood cells don't have it, maybe the brain cells don't have it, but maybe the eggs or the sperm have it. And so that's a possibility. And that's something that Ingrid Scheffer and Heather Mefford and other colleagues of ours have looked at over a large group of kids who have seizures and other problems. And the chance of that happening is something on the order of about 5 to 10%. We used to say it was less than 5%. They've seen some different data now, it's looking at lots of individuals. So I think there's all of that, which is sort of the theory and the drawing out the pedigree. But given the reality of, here's a family and they've got a young child with PCDH19 and now an older child who doesn't have any symptoms, I would say that chances are low for all the reasons we talked about, because all of our groups, we haven't seen many children over the age of 2 present with seizures, but it's not zero. And so you could do testing of a sibling. And, why would you do that? It might actually make a difference in how you treat them. In my neck of the woods, Joe, I still do have to justify genetic testing. It's kind of still a huge hurdle for families to go through getting approval from insurance companies, and we have a whole team trying to help them. But I think if our families go through a lot trying to get testing still. So, but in a case like this, I think it is justified, because if that 2-year-old, 2.5-year-old hasn't presented and she has a PCDH19 variant, I'd want to know that before she gets her chickenpox shot. Or before she gets her next round of vaccines, because maybe I would give some medication around that and try to prevent a fever if I knew she had a variant. And if I knew she didn't have a variant, the power of that negative information for the family to be able to know, okay, we've already got one daughter we're worrying about a lot, at least, we've got to worry about everything else that there is in life to worry about, but at least we don't have to worry about PCDH19 in the other child. That negative information is quite powerful to have if you could have it, and just knowing that you could have it and not doing that is enough reason for me to do the testing.
Great. There's a question about sleep. Several parents in the group, kids don't sleep. I'll take a quick stab at this, and then I may ask Paul how the mice sleep. Sleep is something I almost stop asking any of my epilepsy patients how they're sleeping, because I already know the answer. It's not good. So this is something that's not unique to PCDH19. It's something about childhood epilepsy. And the reason why I joke that I don't ask is because we don't have a lot of great treatments, right, for sleep. Melatonin's mentioned in the chat. Yes, sometimes that works. It's fairly benign. But I'm curious, Paul, in the mice, are there any changes in their sleep behaviors?
We haven't noticed any, Joe, although we haven't had a really sort of proper comprehensive look at it. The mice, as I talked about before, but just in general, they don't have overt seizures,
right?
Not at least that we can see, but clearly their brain activity is elevated by spike wave discharges. So they do have a connectivity defect, if you will, but it's not manifested as obvious seizures. Their sleep pattern, I think Gian Ratto in Pisa has had a closer look at this. I haven't seen anything published, but I know he's looking into this issue. I think he had some evidence that there was a different type of spike wave discharge type pattern in the mice during sleep, but it's unpublished, so I can't comment anymore.
Okay. And then a question about theories as to why children with earlier onset epilepsy may have more serious cognitive delays. I don't know, Joseph, you had mentioned those 2 mutations. Are there anything specific about those 2 mutations that might predict disease severity? I can just say, more broadly speaking, again, in the early life epilepsies that Anne and I both are interested in, which we define as epilepsy onset below 3, maybe arguably 2, we know that regardless of the genetic etiology, that those kids are at higher risk. And the hand waving theory that we have is that there's so much important and rich development that needs to happen in those early formative years, and that if you're having seizures, that can perhaps get in the way of that. But that is certainly a hand waving hypothesis at best. But I don't know, in terms of those mutations, Joseph, would there be any other theories beyond that?
Really hard to tell. The question here is, do the seizures cause the cognitive and neuropsychiatric associated comorbidities, or are these independent of the clusters of seizures? That's how we approach the question. And that's why I alluded to the fact that some parents retrospectively would have noticed that the child was not necessarily behaving 100% normally because they have other children, even prior to onset of the seizures. One could plausibly speculate, based from other models of seizures, that the seizures can be and are likely independent or of other outcomes like the cognitive or neuropsychiatric, but on the same side, these severe clusters of seizures can make those outcomes worse. So there are, in my opinion, would be independent kind of effects on the function of the brain, but of course the ongoing clusters of seizures can make it worse and likely are making it worse. So definitely should be dealt with high priority.
Great. And there's a couple of questions here just about how early can you do genetic testing on a child, and can you actually even do it as part of pre-planned IVF? I mean, you can do genetic testing in a 1 hour old infant, and the same would apply to IVF. I'm not aware at my institution any families that have elected to go that route. Anne, or any of you heard?
Not for PCDH19 specifically, but for others, severe early onset epilepsy disorders, it is dependent state to state in terms of whether companies will actually do that kind of testing, but it's certainly scientifically possible, and some places are able to get it done.
And here's an interesting genetic question. How often should the genetic testing be repeated to look for different variants or classifications of the gene? That's right up your alley.
Yeah, I'm looking at Joseph as well. I mean, I think it's important in a couple of regards. First, if everything is really classic, it's clustering epilepsy, with illnesses and fevers and other features sort of fit, and the variant is, known to be pathogenic, or maybe it's one that's been studied, or it's a truncating variant, meaning it makes a short protein that's been seen before, or there's a family member. There's lots of reasons why we might feel like we're sure we don't need to keep looking. I think the times when we keep looking and sometimes do look more broadly are when things don't quite fit for one reason or another. Either the age of onset was later than you'd expect, or perhaps there's a parent who has the same variant and the parent is completely fine. Then there's another child with symptoms, and it makes you think, well, maybe and who doesn't have the variant, makes you think maybe there's something else going on. So we do occasionally, what we can do now, as Joe was saying, testing has become so much more sophisticated. What we have done on a couple of occasions is to actually say, look, we have a PCDH19 variant, and it kind of fit at the time, but we have some lingering questions. What we could do today is exome sequencing. Let's look to see if there are other and better causes. I know Joseph's been working with one of the families who's seen locally by us in Boston to try to work through a question like that. And in some of my practices patients, we've done that because things didn't quite fit together. We haven't necessarily found alternative causes, but it's one thing to consider. So that's doing more testing when you're not sure if the variant answers all of the child's conditions. The other is just to have the variant reclassified, which is what was mentioned. You can call up a testing company or ask your physician or genetic counselor to call up the testing company and say, hey, is there anything new? Back in 2015, we had this variant classified as a variant of uncertain significance. Has the world learned anything that can actually be brought to bear, and would you call it something differently now? And because of the classification efforts ongoing with ClinGen and ClinVar, there's a lot more catalog now, but the companies aren't necessarily going back and saying, oh, you were tested in the past, let's relook. You have to ask them, but you can trigger that and they can take a look, and they'll do it. Most companies will do it every year or two if you ask them. So that's worth doing, and it's just something that when you go to your next visit, you can ask your physician or ask a genetic counselor, hey, can you take a look? It's just a phone call and they'll do another look.
There's the question about puberty that, why in some girls do the seizures slow down or go away, and why can they and others get worse, so why that variability? I'm not sure if Joseph, the allopregnanolone mini puberty hypothesis, right, in terms of when these girls, usually girls, present when they do, and then go into this more dormant phase of low allo and then peak again. But any theories as to why, even in that big survey, the sort of quiescence or remission was still in a minority?
We have published a graph where we look at how the hormones protect us kind of in the first 6 months of our life, which is called the mini puberty, and that's variable individually. Some individuals have this mini puberty lasting almost 8 months, up to 1 year. Other individuals have this mini puberty in the first 6 months, and then they go into low hormone levels until they go into the puberty at around 10, 11, or sometimes later. So we have superposed the onset and offset of all girls' information which we could collect. And it really fits perfectly that as soon as we see the dip of mini puberty, we see really huge rise in onset. And as we're going into the puberty, we really see the offset. So that fits perfectly. Now, why we have these outliers, why I still trying to bend my brain, why there would be some girls who would actually go worse during puberty. Maybe the answer is in the technologies the Marinus is using, and it's not easy to get hands on it, and that is really testing for the neurosteroid biomarkers to see what is the baseline profile of these individuals, because we know it simply from looking around from our friends. My facial hair started at the age of 27. I was a frustrated teenager. How come I don't have a facial hair when I'm 15-year-old? It just didn't happen. I was teased. The first time I shaved, I was 27. So is this, I don't know, it's probably hormonal to some extent. So we are hormonally very different. And maybe the explanation might be there because we know that the hormonal system, it's tampered with. And perhaps the explanation might be there, and that would require more testing for these molecules, which is not many laboratories do it routinely. And we have to say, because these are so-called neurosteroids, so they are produced in the brain, but we still try to measure them in the blood, and that's non-trivial. So we struggle really how best to interpret that information and relay it back into the brain.
A quick question for Alex, actually, about ganaxolone. So when will ganaxolone be approved, and is Marinus still working toward approval as a general seizure medication? Are there options for future drug trials, since PCDH19 seems to be difficult for a medication trial?
Yeah, thank you for that question. So the study that I presented would not support regulatory approval due to the size and the outcome. But as I mentioned, we are looking to publish those data. With that said, we did have a positive study in another rare developmental and epileptic encephalopathy, one that Sharyl mentioned earlier, CDKL5 deficiency disorder. So we are filing an NDA for that in the middle of this year and hopeful for FDA approval sometime next year. But again, that would only be indicated for seizures associated with CDKL5 deficiency disorder. We are looking at other indications as well within our pipeline, where we do think there could be potential benefit as well. And again, I think that, with some new clinical trial design, potentially, I think PCDH19 could be very interesting in the future.
So in our last 5 minutes here, we got a lot of flurry of questions here at the end surrounding AEDs, so anti-epileptic drugs, anti-seizure medications. Is there data to suggest that early use of AEDs disrupts brain development, making it worse? This is a very, very challenging question to answer, so I guess maybe I'll take it. We just don't know, if I'm being completely honest, because we don't put kids who don't have seizures on anti-seizure medications. I think we feel that over the decades of using some of these medications, and more recently some of these medications that do seem to be less sedating, have less drug-drug interactions, that if they are contributing to some adverse cognitive profile, it's minimal. And like everything in medicine, it's about risk-benefit. But my feeling is that we don't want to let these clusters of seizures be left untreated, because I think that could, and this again, just my opinion, could result in a worse outcome. I'm not aware of animal data. The animal data that's done for these they're used in supraphysiologic doses, right? That we generally don't, that to establish neurotoxicity, generally they're tenfold higher than what we use in clinical practice. I don't know, Paul, I see you shaking your head. Do you have anything to comment in terms of animal studies? No, okay. And just in the last few minutes, so would there be a correlation between seizure onset and the X inactivation process in relation to severity? What would be the most likely factor to have a more severe representation of PCDH19 versus a later onset? That's a great question. So maybe what's being asked here is the degree of mosaicism. I was actually going to ask that question to Paul and Joseph. So is there been established that what, we talk about therapies and knocking out, and is there a hypothesis as to what threshold? Is it 20%? Is it that maybe can be still unaffected?
I can tell you my take on it, and it's not, it's my opinion, and there's only limited data to support it. But I don't think it's just the level. It's not just the numbers. You can talk about this exit activation process being 20%, 30%, 40%. I think it's probably going to be much more about where it's occurring within the brain, which pathways, which neural connections are affected by those changes, rather than just the global overall level. Hopefully that makes sense. We're dealing with a three-dimensional organ that in humans has 100 billion neurons. It's going to matter a lot which of the neurons either have predicate here at 19 or which of them don't. So, and that makes this really the million-dollar question for me is this one: what's the connection between the pattern of exon activation and ultimately the phenotype that we see in individuals? That is an exceedingly difficult one to answer, but I think it's one that we have to have a crack at, because that's, I think, the pathway to understanding how it is that the genetic changes ultimately give rise to different phenotypes in different people. And on top of that, if we can understand that question, then perhaps we can have better drug, interventions, or even consider genetic therapies on top of that as well, which I'm happy to talk about, but perhaps there's not enough time. But yeah, I think it's a really, really good question, but it's just very difficult one to answer because, as I said before, each brain is unique to that individual, and the pattern of exon activation will affect how the phenotypes play out.
All right, well, we're running out on time here. Go ahead, Joseph, you want to say one more thing?
Just very quickly, we look at exon activation from different available tissues from patients, like from blood, saliva, skin, and that was not informative. So Paul is right, and we can't sample brain. But what is really interesting here, exon activation is in fact epigenetic modification of gene expression. We see different gene expression also, and nicely showed it in her presentation in zebrafish, that this mosaicism kind of changes how the cells express different other genes. And that is also governed by epigenetic modifications similar to exon activation. And we just try to look into that instead of just exon activation to see whether that's not going to tell us a little bit more about why some heterozygous individuals, whether they are mosaic males or heterozygous females, are affected more severely than the others. So I think there is a way, I think there is a hope, because similar tests are available for about 60 other neurodevelopmental disorders and with seizures, where we can kind of read the impact of the loss of function of that gene on the level of the epigenetic modification, and exon activation is one form, quite frequent, of such modification.
Okay. And I want to answer one last clinical question here, and then I'll end on a nice, happy comment in here. So there's my 9-year-old daughter with autism and uncontrolled seizures presents with episodes where she, one moment to another, becomes lethargic, sick expression, downcast eyes, responds late to orders, her temperament and mood change. It's as if his body, mind lost their strength. After minutes or hours, she returns to normal, face is happy again. What could be happening? So I'll take a stab at this one. So, we know that seizures can do anything, and we sort of define as a concern for a seizure as being something that's repetitive and stereotyped. If this is repetitive and stereotyped, we do see these odd things in Dravet syndrome patients that we call this obtundation status, where just kids are just acting different for a period of hours, and then they sort of come back, and sometimes they even come back after they actually have a more typical seizure. So again, without knowing your child specifically and other things that are going on, I do feel it's very likely related to the PCDH19, and it may be a seizure or seizure phenomenon. And I can have Julie connect me with you via email afterwards, and we can maybe share a little bit more details. But I want to end on one other sort of, it kind of brings an answer to a question sorry, I'm trying to find it, went back. So it's from Meredith. I was 9 years old when I started having seizures and was just diagnosed with PCDH19. My seizures went away with my last pregnancy, and I've been off medications for 3 years. So I think that this is obviously something that we hope for more and more of our patients, and it's nice to know that this can happen. Thank you all for your questions and for coming. I apologize that we couldn't see everyone's faces for our technical glitch here. But like Julie said, as soon as we can all get together in person again, we will have more time to hear more of this great science and to just get together and share stories. So thank you to our panel from across the globe, and everyone stay well, and we'll talk soon. Julie, any closing comments?
No, just thank you, everyone. It was a really engaging and informative conversation, and I want to thank everyone for making the time to be here.
All right, have a nice weekend, everyone.
Take care. Hope we're in person next time.